Thank you, Mr. Chairman, and good morning, everybody. I'm going to present a study developed at the Magna Graecia University of Catanzaro, focused on the design and fabrication of a pyroelectric sensor for induced pluripotent stem cells characterization. The topic covered by this talk will include a brief introduction on the problem of cell line characterization for retrieving information from single and multiple cell samples. Then, we will discuss the main aspects of the proposed sensor based on infrared-induced pyroelectric effect to detect thermal differences in liquid droplet samples at different cell concentrations. In particular, thermoelectric transduction will be evidenced as a way for providing thermal characteristics of the cell sample. Finally, the fabricated prototype and experimental results on induced pluripotent stem cell obtained from patients affected by Fabry seizures will be presented before to conclude. Cell characterization is a repetitive and common task performed in molecular biology and clinical laboratories on different biological fluids. It provides information about, for example, concentration, proliferation, and viability, ensuring reproducibility and accuracy during in vitro experiments. Among the most diffused and common technique, cell counting is performed every day using manual and automated counters. Flow cytometry, which is a gold standard for cell characterization, is based on hydrodynamic focusing and light scattering of each cell. Low-angle scatterings provide information about geometry, diameter, volume, etc., while high-angle scattering provides information about internal properties of the cell. Another widespread automated cell counter is based on the Coulter effect, in which cells induce a ionic current variation flowing between two electrodes in electrolyte solution. Also, colorimetric assays, whose protocols are based respectively on the use of dyes, are daily used for cell viability assessment. Actual ongoing investigations are thus oriented on improving retrieved information versus smaller, more portable, or even wearable devices. Recently, thermodynamics of cell aroused considerable scientific interest since it has been involved in a variety of biological mechanisms. Living cells can change their temperature during cell activities and also under external stimuli, leading to acute variation of intracellular temperatures from the normal state. However, such temperature change inside cells is usually at a small scale and is of transient nature, and is rather difficult to be measured. Despite different nanothermometers were developed evidencing intracellular temperature spikes, many controversies still exist along the published data, especially in terms of thermogenesis using temperature as a probing index. Thermal conductivity was recently proposed for the evaluation of cell viability, and in particular, the thermal conductivity was related to the death mechanism and thus proposed as probing index of cell viability. Hereafter, we provided a pyroelectric sensor which relies on the thermal conductivity of a single cell. Among cells, induced pluripotent stem cells are obtained by reprogramming terminally differentiated cells. Like embryonic stem cells, iPSCs have the potential to differentiate towards all three germ layers, ectoderm, mesoderm, and endoderm, retaining the same genome of the donor. For this capability, iPSCs represent a useful tool for model disease, drug screening, and regenerative medicine in a patient-specific manner. The fundamental of the proposed solution is based on the use of polyvinylidene-fluoride, a ferroelectric polymer, and in particular exploiting its pyroelectric properties induced by infrared radiation. As shown in figure, a PVDF layer is interposed between an infrared source and cell sample. It is coated with aluminum and graphite and is placed in thermal contact with the sample. Infrared source at about 850 nanometer is used to stimulate the pyroelectric element. Charge generated by the PVDF is acquired and processed by the front-end amplifier designed as a charge-sensitive amplifier topology. The sensor has been designed to be as minimal as possible, avoiding, for example, microfluidic components and reducing the fluid handling in just two simple operations: the pipetting of a sample into the well and its washing with the same procedure. The well dimension, including this transducer, is about half centimeter. The sample volume has been established in 25 microliter with a liquid height of about 1.3 mm. A higher surface contact is preferable to minimize lateral to base surface ratio. By the thermal point of view, the layered structure composed by the PVDF and the liquid sample can be modeled through their respective heat capacity and thermal conductance. The latter represents the heat exchange via heat conduction and radiation. In this slide is reported the thermal mechanism of the pyroelectric sensor. As stated before, the sample and the PVDF are characterized by heat capacity and thermal conductance. We assume that the absorption of radiant flux causes homogeneous temperature distribution in the transducer, creating a difference in temperature between the lower and the upper surface of the PVDF. Considering the equivalent thermal model reported here in the slide, for a temporally constant radiant flux, the difference in temperature across the transducer is related to the radiant flux itself and to the overall thermal conductance. In the actual arrangement, the sensor has been designed in order to verify the reported conditions, and thus the variation in temperature across the transducer is directly related to the thermal conductance of the sample. In this work, we used iPSCs obtained from patients affected by febrile seizures. This convulsive event happens in some children during fever and can be related, as in our case, to genetic mutation in the gene SCN1A, codifying for the voltage-gated sodium channel NAV1.1. We dissociated human iPSCs colony using the reagent acutase, a solution of proteolytic and collagenolytic enzymes, to obtain a single-cell suspension. Sample concentration was evaluated through a calibrated chamber. Analyses were performed using 5 serial dilution of the cell suspensions. The pyroelectric response, as reported with the red line, is characterized by a fast rise time due to thermal properties and a slower decay, which is due to the charge amplifier topology. The parameter evaluated during the experiments was the area under the curve, AUC, that is the integral of the sensor output evaluated during infrared stimulation, as you can see in the shaded area. The goal of our work was to test whether a single missense mutation in the SCN1A gene could cause variations of the thermal characteristics of a mutated cell respect to control cells. In this slide, we reported the results obtained by the analysis of 25 microliter of iPSCs samples from healthy patients and patients affected by the mutation. As reported in the upper hand side picture, as cell concentration increases, sensor output increases accordingly. In particular, the pyroelectric effect is greatly influenced by a reduction of thermal conductance of the sample as a consequence of the presence of more cellular bodies inside the sample. However, here in the slide, differences between the samples is evidenced. To this end, the thermal conductivity can be related to the area under the curve by considering the physical and electrical characteristics of the sensor and by considering the Maxwell model, which describes the thermal conductivity of a solution in which spheroids are dispersed. After this analysis, in the lower figure is reported the related thermal conductivity, in which iPSCs affected by the mutation showed a statistically significant increase in the thermal conductivity with respect to healthy control. So the AUC increases by increasing the sample concentration. These variations are caused by variation in sample thermal properties. At the same time, the healthy control evidenced a lower thermal conductivity respect to the mutated cell samples. Summarizing, in this talk we presented the pyroelectric sensor for iPS cells characterization. Thermal behavior and experimental results on cells from healthy patients and patients affected by SCN1A mutation has been presented and compared. Preliminary analysis from our study showed a statistically significant increase in the thermal conductivity of mutated cells respect to healthy control. These data provide evidence on how single mutation can substantially affect the physical characteristics of a specific cell. The proposed sensor results thus promising for the development of an alternative technology to be used in molecular biology and clinical laboratories for the thermal characterization of cell line. Thank you for your attention.